Howdy, today on Flipping Science we're talking about organelles. So here's the context that we're exploring. We're going to review or study the concept of the organelles, and we're going to use some examples. We're going to pretty much go through all these examples here and we'll talk about them. So what is an organelle? So an organelle is a structure inside a cell that does a specific task. If we look at our picture here, we can see several different organelles. Of course, they're never arranged like this. This is just to make it easier to see what's going on. We can see the nucleus, we can see the endoplasmic reticulum, we can see Golgi bodies, we can see mitochondria. So there's several different organelles in here, and we're going to talk about some of those. So the first organelle we're going to talk about is the nucleus. So here is my cell model. We can see the nucleus is the big round thing in the middle. We've already talked about the nucleus a little bit. It contains the DNA. So it's very important for holding the DNA for most of the cell's life cycle. The DNA is important because it tells the cell what to do. The DNA contains the instructions that the cell follows in order to grow, divide, reproduce, access energy, and so on. So the nucleus is very important. And it's, again, I've said before, it's the most easily recognizable structure inside the cell because it's big and round and it's right in the middle and it's a bit darker. So ribosomes take information from the DNA in the nucleus and turn that information into proteins. So here's a little picture showing down here. This is called translation. So a copy of DNA is made on a molecule called mRNA. The mRNA is read by the ribosome, and little molecules called tRNAs that hold amino acids come in. They drop off their amino acids, the amino acids are joined together, and then they fly away. The chain of amino acids together makes a protein, but you can have several amino acid chains joining together to make a larger protein. Here's an electron micrograph showing the structure of the ribosome. There are these very small dots in here, so they're very, very, very small. Ribosomes are made up of two subunits. There's a large subunit and a small subunit, but like I said, both of these are very, very small. Key things about ribosomes: they're not membrane bound, and they are found in both prokaryotic and eukaryotic cells because all cells need to make proteins. Another organelle is the mitochondria. If we look on my cell model here, these things down the bottom, they're the mitochondria. The mitochondria used to be bacteria that were free-living. They moved inside eukaryotic cells a while ago, and their job is to give off energy from food. So we often refer to them as the powerhouse of the cell. So they use oxygen from the air that's taken into the organism and into the cell, and they use the oxygen to help break down sugars and release energy from sugars. That's the job. So they're very important. The way I remember what a mitochondrion looks like is it's got this kind of maze-like structure in the middle, so it looks like a maze. So as I just said, mitochondria are responsible for a process called aerobic respiration, which is where you use oxygen to break down sugar, so glucose is our sugar here that we're looking at, and you produce carbon dioxide and water as byproducts. Now I'm going to do a little bit of a quiz for your chemistry people here. I want you to try and figure out what numbers are going to go in front of the oxygen here, the carbon dioxide here, and the water here. So we'll see how you go in a second. So if we look here, we've got 6 carbons on this side, and we've only got 1 carbon on that side. So we're going to need at least 6 here. So we put a 6. If we look at our hydrogens on this side, we've got 12 hydrogens. Over here we've got 2, so we need to times 2 by a number to get it up to 12, and that number is 6 as well. And then if we total up our oxygens on this side, we've got 6 times 2, so that's 12 oxygens, plus another 6 is 18 oxygens. And on this side at the moment we've only got 6, so we need to have another 12. So we need to have 6 times 2, so 12 oxygens. So this is a handy equation to learn. The whole point of this, the reason why I'm talking about this is why we're breathing. We breathe in oxygen in order to break down sugar to get our cells producing energy, and we use that energy to do all those processes like move around, make new chemicals and so on. So getting energy is very important. The next organelle we're going to look at is the chloroplast. So in my plant cell model here, the chloroplasts are these kind of green blobby things down the bottom there. The job of the chloroplast is almost the opposite of the job of the mitochondria. It's making sugar that we can then burn later using mitochondria. So the chloroplasts, they take carbon dioxide and water from the environment. They use light energy to break up the water and the carbon dioxide to make sugar and oxygen as a byproduct. So they're very, very important. The way to tell if it's a chloroplast is it's green, and normally it's got these stacks of coloured bits in them called thylakoids. So a stack of thylakoids together is called a granum, and when you look at them, you can see this kind of bandy-like appearance inside. Chloroplasts are ridiculously important because if you think of the food you eat, most of the food you eat is either a plant or something that ate a plant or something that ate something that ate a plant. These guys generate the energy for plants using sunlight, carbon dioxide, and water. The other thing they generate is oxygen, which is handy because we breathe out oxygen. The endoplasmic reticulum, it's a system of membranes that extend from the nucleus out. Their job is for transport. There's two types of endoplasmic reticulum. There's rough and smooth. If we look on our plant model here, we can see the endoplasmic reticulum going around the nucleus there. Now, there's ones that have dots on them. You might not be able to see that. That's the rough endoplasmic reticulum. And there's ones that don't have the dots on them. They're the smooth endoplasmic reticulum. The dots are ribosomes. And we talked about ribosomes before. They make proteins based on information given to them by the nucleus. So the rough endoplasmic reticulum contains proteins that can get made at the surface of the endoplasmic reticulum. Then those proteins can be packaged and sent off. The smooth endoplasmic reticulum, they don't have any ribosomes attached. Their job is they help with metabolic processes, they help to make lipids, and they also help a little bit to break down carbohydrates as well. So oftentimes they have enzymes embedded in the endoplasmic reticulum that are too small to see. Two types of endoplasmic reticulum, rough and smooth, both involved in transport and synthesis and making things. But the rough ones have ribosomes attached. Next organelle we're going to talk about are the Golgi bodies, or they're sometimes called Golgi apparatus. Very important when you're naming them, you need to have a capital G for Golgi because it's named after Mr. Golgi. We're going to talk about him a bit later on. The Golgi bodies, they're flattened stacks of membrane. So in my plant model here, the Golgi body is this brown one. So that plant cell, we can see the Golgi body in there. It's involved in packaging and secretion. So it can take in proteins that are produced by ribosomes, package them into little vesicles, which are these little round bits of membrane essentially, and those vesicles then go to the cell membrane and get spat out. So the internal contents of the vesicle get spat out into the environment. So they're very important for packaging up materials inside the cell, but then also secreting them to the outside environment for the rest of the organism to use. They're involved in a process called exocytosis, and we might talk about that a bit later on. So today I've been switching a bit between my animal cell and my plant cells. Plant cells have some different structures to animal cells, but they share some that are similar as well. One of those structures is the cell wall. So on my cell model here, if you look at the edge, there's kind of a lighter color on the inside and a darker color on the outside. The lighter color is the cell membrane, which is on the inside, and the dark color on the outside, that's the cell wall. Now, plant cells and fungal cells also have cell walls. We're not going to talk about fungi in too much detail. The cell wall is important for maintaining the shape of the cell. So if there's lots of water or little water, the shape of the cell kind of maintains itself. And we use those cell walls. If you're writing on paper right now, you're writing on the cell walls of plants. That's what paper is made up of. It's made up of cellulose. Another big structure you get inside plant cells is vacuoles. If you look at my plant cell model here, we can see the big green vacuole down the bottom. It's a big storage space. It can store water, it can store salt and sugar as well. And the plant cell can pump water in and out of the vacuoles to change the shape of the cell. So we get water going in and out, and that can make the cell swell up or shrink down, depending on what the cell's trying to do. This is how plants can move. So if you look at some plants that follow the sun, for example, sunflowers, the way they're moving is they're pumping water in and water out of a particular cell so that they can move around. So if we go back to our animal cells over here, animal cells don't have that large wall around it. They don't have a cell wall. The other thing is that they don't have large vacuoles on the inside. They do have some small vacuoles that they can use for storage, but they're not as large as the ones in plant cells. And obviously, like I said before, they don't have chloroplasts. They make their energy just from the organisms that they eat and consume, and then their energy is released generally by the mitochondria. So they don't have to make chemicals that they then break down to get energy out like plant cells do. Because animals by definition have to consume other organisms to survive, they get their energy from there. So they don't have any chloroplasts. So while we're talking about this, it's important to note also that both plant cells and animal cells do have mitochondria. The mitochondria break down chemicals to release energy. Plant cells, they make chemicals with extra energy by using photosynthesis. Then they break those same chemicals down using mitochondria to release energy. So they contain mitochondria and chloroplasts, whereas animal cells only have the mitochondria. So let's look at some science as a human endeavour here. we'll talk about Golgi bodies. This is Mr. Golgi over here, and I love his huge mustache. He discovered Golgi bodies when he stained cells that he was looking at under just light microscopes. He stained cells in a particular way, and he saw the structures of Golgi bodies, but a lot of people didn't really think that it was a structure inside the cell. It was just kind of an artifact of his staining technique. So he argued back and forth with other people for a while, but it was really only when the electron microscopes came in the 1940s and 50s that the existence of the Golgi bodies was confirmed, and also their function was figured out. So people could see the vesicles coming in and coming out of the Golgi bodies, and they determined the function from there. So again, we're seeing the effect of technology in biology by going from a light microscope that Mr. Golgi was using to the electron microscopes that others were using. We got more information and we could see more developments. So it's that link between technology and biology, again, leading to advancement of the science. So today on Flipping Science, we looked at some organelles found in animal and plant cells. That's it for Flipping Science today. See ya.